Hello, Biology 3270, 5270. Today we are going to continue talking about the carbon and nitrogen cycles, and we will continue to use the ocean as our prime example. Specifically today we're going to focus on oxygen deficient zones of the ocean and the fun ways that the nitrogen cycle plays out in those regions of the ocean, and I'll explain what oxygen deficient zones are when we get there. First, let's use my beautiful cartoon of the ocean as our working model of how the carbon and nitrogen cycles are interacting with each other. We've talked about how the green phytoplankton in the surface ocean are fixing carbon dioxide into biological carbon. Oxygen is the byproduct. And that this organic matter can eventually be exported to the seafloor when the phytoplankton die in big chunky mats, or when animals come by and eat the phytoplankton and the other microbial consumers of the phytoplankton and poop them out. And the fecal pellets containing this organic matter sink because they are heavy and big, and that is a very important means by which carbon is exported to the seafloor. As this carbon is consumed by microbes primarily and converted back to carbon dioxide, and we talked about when this happens in the surface ocean, that's called the microbial loop, because the carbon comes in as carbon dioxide, it gets fixed, and then it goes back out as carbon dioxide again when heterotrophic bacteria consume that matter. If there is more carbon than oxygen in a particular region of the ocean, or a particular depth in the ocean, then you run out of oxygen before you run out of carbon, and that's when you get anoxia. You get regions of the ocean that do not have oxygen, but you still have organic carbon. And those are the regions of the ocean that we're going to talk about today. So if you run out of oxygen, then the electron tower predicts what is the next best electron acceptor to use in that situation. And that would be nitrite or nitrate, right? So if you're going to use nitrite or nitrate and nitrate are very similar to each other. Their redox states are very similar. The minor differences between nitrate and nitrite are important if you study the nitrogen cycle. Today we're mostly going to treat them as one thing, but we'll occasionally mention when they get converted between each other. Okay? But if you use nitrate or nitrite as your electron acceptor, you can say that you're breathing it instead of oxygen, and you are consuming organic matter. We'll use glucose as our example of organic matter. Then you are doing denitrification. That's what that is called. And we had previously mentioned this when I went over the electron tower, that in a lake and ocean mud, you see the electron tower in action, that in the top of the column of mud, oxygen is consumed first. When the oxygen is gone, then you get nitrate being used as an electron acceptor, and that is denitrification. So first you have aerobic heterotrophy, then you have denitrification, which you can think of as heterotrophy with nitrate. That's basically what denitrification is. It's heterotrophy with nitrate instead of with oxygen. And below that, you use these other electron acceptors that we can talk about another time, although we've previously mentioned methanogens that use CO2 as their electron acceptor. Okay, so this is what denitrification looks like in my cartoon of the ocean. There's no more oxygen, so you're using nitrate as your electron acceptor, and the byproduct, the waste product of denitrification is into gas, or at least eventually it can result in into gas, as we will talk about later. Into gas is a gas, so eventually it can escape the ocean, basically evaporate back into the atmosphere. So denitrification is an important nitrogen removal process. It is often referred to as nitrogen removal in an ecosystem context because it removes aqueous nitrogen from the water and puts it back into a gaseous state where it can escape to the atmosphere. So nitrogen is actually removed from the ecosystem. Don't forget that this is different from nitrification. They are very similar words to each other, only differing in the DE before denitrification. And that prefix of D before denitrification is very important because it indicates that it is the opposite of nitrification. So nitrification and denitrification are the opposites to each other in every way. Nitrification produces nitrate, which is what is consumed by denitrification. Nitrification is autotrophic. I mentioned last time that nitrifying archaea and some nitrifying bacteria fix CO2 as their carbon source. They are autotrophic. And the electron acceptor for nitrification is oxygen, so it's aerobic. Denitrification is heterotrophic. It is consuming organic matter, right? And it's anaerobic, obviously, because it's using nitrate and not oxygen. So denitrification and nitrification are the opposites to each other in at least these three different ways. And you can see that in this diagram where nitrification is up here in the oxic zone of the nitrogen cycle, and denitrification is down here in the anoxic zone of the nitrogen cycle. All right, let's say a few more words about denitrification. First of all, as I hinted before, denitrification actually requires many steps. You are taking nitrate, which is the most oxidized form of nitrogen, all the way to N2, which is almost the most reduced state. It's not as reduced as ammonium, but it's still highly reduced. And it goes each step of the way. So from nitrate to nitrite, from nitrite to nitric, from nitric oxide to nitrous oxide, and nitrous oxide to N2. Each step of this pathway has to be catalyzed by different enzymes and generally by different species of bacteria. Some species can do multiple steps, but some species can only do one of these steps. The final step is a particularly interesting example. It's catalyzed by an enzyme called nitrous oxide reductase. It's reducing nitrous oxide to produce N2. And it requires 12 copper atoms at the core of every single enzyme. So every single one of the enzymes in the bacteria that are doing this step requires 12 copper atoms at in the catalytic core of the enzyme. And that's really a lot of copper. That's a lot of metal for one enzyme. And when copper is limiting, so if you grow these denitrifying bacteria in conditions where there's not much copper, then they can't do this final step because they need so much copper for this enzyme. And they stop at nitrous. They don't make N2. And that's pretty important because nitrous oxide is a very powerful greenhouse gas. So if we produce more nitrous oxide into our atmosphere instead of N2, then the Earth will get warmer because nitrous is a good greenhouse gas and N2 is not. So it's important to understand this process, if only for that reason. And perhaps a more interesting, more exciting, exotic way to look at this. Greenhouse gases are obviously important today, but you may not have thought about the importance of greenhouse gases 1 to 2 billion years ago. So I thought I would mention this fun implication of nitrous oxide reductase. So here's a figure from a really fun paper, really short too, 1 or 2 pages, in 2007. By Roger Buick, who I met when I was a student in the astrobiology program at University of Washington, where he is talking about the Proterozoic era, the Proterozoic being what is often referred to as the boring billion, this time between 1 and 2 billion years ago, when not a whole lot was happening compared to other periods in Earth history. So the most recent 1 billion years has been the time of the diversification of all the animals and plants and life as we know it. From 4 to 3 billion years ago, Earth was, becoming hospitable. You have liquid water, you have continents forming, you have lots of really fun microbial activity going on. The first evidence of life, really important steps in the early evolution of life from 2 to 3 billion years ago is when the great oxygenation event occurred, where you went from an Earth having no oxygen to an Earth having almost as much oxygen as we have today. And that must have been a lot of really fun biology going on there with such a dramatic transformation of the planet. And then from 2 billion years ago, you have basically life as we know it now. It's an oxic world. You don't have animals and plants yet, and you just have a lot of microbes hanging out for 1 billion years. Almost certainly there's more going on there than that, but that's why it's called the boring billion. Okay, so during this boring billion of the Proterozoic era, due to a variety of factors of ocean chemistry and atmospheric chemistry, it was a tough time to be a metal in the ocean. There was a decreased solubility of metals in the ocean at that time, in part because there was an increase in hydrogen sulfide at that time. So we were producing more sulfide during this period, and hydrogen sulfide will precipitate out with metals and form metal sulfides, dropping them out of solution. And there are also some other reasons why the concentrations of these metals were expected to be lower during this time than they are now, and also even lower than they were before then. So anyway, the bottom line here is that we think that copper was at its lowest concentration in the history of the Earth during this time. It was so low that it's possible that it was limiting for nitrous oxide reductase. It's possible that all the denitrification in the world was not able to go all the way to N2, and you're producing a lot of nitrous oxide. And you may have had a whole atmosphere full of nitrous oxide, and you may know that nitrous oxide has another name called laughing gas. And so it's just kind of fun to think of a whole world full of laughing gas. And so instead of the boring billion, maybe it was the funny billion, the laughing billion. Okay, so that's just a fun aside about how these things might have played out a long time ago. Let's come back to the nitrogen cycle because we have one more player in the nitrogen cycle that we need to talk about. We talked about denitrification, we talked about nitrification, and now we're going to talk about anammox. Anammox also produces nitrogen gas, just like denitrification. It also starts from nitrite, although not nitrate. But instead of organic matter, instead of being heterotrophic, it uses ammonia as its electron donor, and it's an autotroph. It fixes CO2 to make its own food. So it takes ammonia, nitrite, combines those together to form N2. So you can think of it as an alternate and also very weird pathway for denitrification. It is not denitrification, it has a different name, but it also produces N2 starting from nitrate, so it has similarities to denitrification. And for the next few slides, I'm going to compare and contrast anammox and denitrification and really dig into this into some detail. The process of anammox is only carried out by bacteria in one group of the bacteria, in phylum Planktomycetes, and even one particular group within the Planktomycetes. This group of bacteria, the Planktomycetes, were already known to be among the weirdest and most unusual of all of the bacteria, even before it was discovered that they could carry out anammox. And one of the reasons why they were weird is because they were known to have various kinds of internal cell compartments. And for a time, they were even thought of as potential missing links between prokaryotes and eukaryotes because they have these cell compartments. I don't think that idea has a lot of support today. Now we just think of them as weirdos. Because we don't think these internal cell compartments are evolutionarily related to the internal cell compartments that eukaryotes have. But I think that idea still has some proponents even today. All right, so these Planktomycetes that do anammox have a cell compartment inside their cells called an anammoxosome, where they carry out the anammox process. The lipids that form the membranes of these anammoxosomes are made of a lipid structure that is called laterane. Here is the enzymatic process of anammox. If you want to learn about that, I won't mention these details anymore in this lecture. But these laterane lipids are really weird. They're made out of squares, essentially, these parts of the lipid molecules. And they sort of look like ladders, which is why they're called laterane lipids. And the idea is that these square-shaped lipids should pack together very, very tightly and should be essentially leak-proof, not enabling anything to leak in or leak out. And my understanding is that the idea is you want to have a leak-proof anammoxosome because an intermediate in the anammox process, which is listed here, N2H4, is hydrazine. And you may have heard of hydrazine before because it is the primary component of rocket fuel. So a intermediate of the NMOX pathway is rocket fuel, and that is perhaps why they need these really weird lipids to keep the rocket fuel inside the NMOXisome and not leaking out into the rest of the cell. This is all really pretty new research, and I think there's still a lot that we don't know about it. But those are the important points of what we do know so far. Okay, so let's go back to the ecology of how this works, how these different pathways interact with each other in an ecosystem. So again, here we have denitrification. It's fueled by organic matter being pumped from the surface to the bottom. When there's no oxygen, nitrate becomes the best electron acceptor. You produce N2 by consuming organic matter. The NMOX bacteria are autotrophic. They do not need the organic matter, and they still produce N2. Okay, now where this happens, again, is where you run out of oxygen. Typically, that happens in sediments, where the oxygen is consumed very quickly in the first few centimeters of the sediments. And that's because the sediments are packed so tightly together that oxygen can't diffuse faster than the microbes can consume it. Whereas in water, oxygen diffuses faster, and so you have to consume the oxygen much, much faster to deplete it in water than you do in sediments. So it's very easy to get anoxic conditions in sediments. And so most of the nitrogen removal processes, that's the general term for denitrification and NMOX together, because they're both removing nitrogen. Most of NMOX denitrification occurs in sediments because it's easier to find anoxic conditions there. Okay? But there are places where you do consume the oxygen matter fast enough that you can consume the oxygen fast enough that it's faster than diffusion, and you can get local regions of anoxia where the oxygen has run out. And we call those places oxygen deficient zones. That's a fairly self-explanatory term. You have zones where the oxygen is deficient. And in those places, you can get denitrification and NMOX occurring in the water column at some depth just below the surface, and you don't have to wait. They don't have to wait for the organic matter to get to the sediment before they can act. You can find them in the water. So here is an explanation of where in the world you find these things. Oxygen deficient zones are also called oxygen minimum zones. There's a minor fight in the community about what is the best name for them. So these are natural features. They've probably always been on Earth. However, it seems increasingly likely that humans are making oxygen deficient zones grow and perhaps get more numerous. I'll show a slide indicating that in the next slide. You tend to get them naturally in coastal areas, in the same places where you have really productive fisheries, for example, as I was talking about in the previous lecture, where you have upwelling, bringing fresh nutrients like nitrate up to the surface, fueling a very productive ecosystem with phytoplankton, fixing carbon, creating more organic matter, animals coming in, eating that organic matter, creating a nice productive fishery. All of that dead phytoplankton and fish poop gets consumed. And if there's enough fish poop and dead phytoplankton, you can deplete all of the oxygen just below that highly productive fishery, and you get fun anaerobic microbial processes like denitrification and NMOX because of that confluence of conditions. The upwelling itself actually acts to accentuate the oxygen deficient zone because the upwelling water is actually pushing the water up and preventing this oxygen deficient zone from sinking and spreading out and becoming more diffuse and mixing with the rest of the ocean. So the upwelling is not only bringing fresh nutrients to support a more productive ecosystem at the surface, it's also acting to trap that oxygen deficient zone at that relatively specific depth just below the surface. Estimates have large error bars because we've only started studying these oxygen deficient zones relatively recently, and they do seem to be changing, and so our understanding about them is also changing. But we think about 30 to 50%, so maybe up to half of all of the nitrogen removal, all the nitrogen loss, in the oceans does occur in the water column and not in the sediments. So just a little bit on whether humans are causing these oxygen deficient zones to grow. Here is mostly a modeling study, I believe, using some real data and doing some simulations in these different zones of the ocean. So these 6 different panels are showing 6 different locations in the ocean here. And you can see that from 1960 to the present, it seems like the degree to which these oxygen deficient zones are deficient in oxygen, so the concentration of oxygen seems to be decreasing, so more blue colors means less and less oxygen. And the depth over which you find the oxygen deficient zone seems to be growing in at least most of these locations as well. Some places where you may not have thought that the oxygen had decreased to a level such that you would call it an oxygen deficient zone, in the 60s, 70s, and 80s is now getting to the point where you actually have real anoxia. So in these oxygen deficient zones, we have both denitrification and anomox occurring. But in different conditions, at different times and different places, you might have more denitrification or more anomox. And our understanding of when denitrification versus anomox is favored in a given time or place is constantly changing. We've only discovered anomox relatively recently, and so this is really a new research topic. And this kind of competition, or perhaps cooperation also, between denitrification and anomox is really important to study because it has some far-reaching implications. So, for example, depending on whether denitrification or anomox is more dominant, it affects whether we think that the loss of nitrogen from the ocean, so whether nitrogen removal is dependent on autotrophic or heterotrophic activity, right? Because denitrification is heterotrophic and anomox is autotrophic. That's a pretty big difference. Also, do we think that the loss of nitrogen depends on a single kind of bacteria, these weird anomox bacteria and the Planktomycetes? Or does nitrogen removal happen basically no matter what the ecosystem looks like, because lots of different kinds of bacteria can do denitrification, whereas only one specific group of bacteria can do anomox? And then it also has implications for whether pumping carbon from the surface to the deep ocean is a source of leakage of nitrous oxide. Denitrification has this nitrous oxide intermediate, and so whenever denitrification is occurring, you're always going to have at least some and maybe a lot of leakage of nitrous oxide, which is important to know about, whereas anomox does not have nitrous oxide as an intermediate. So anomox does not produce nitrous oxide. And so there are some pretty important differences, and here are just 2 of the papers that came out kind of late 2000s, early 2010s, where there were some really high-profile studies asking these questions, and different groups got different answers. So here is a paper where the title is Denitrification as the Dominant Nitrogen Loss Process in the Arabian Sea. So denitrification is more important than anomox. And here is a paper with the title Revising the Nitrogen Cycle in the Peruvian Oxygen Minimum Zone. So here they're going off the coast of Peru. Here they're going off the coast of India in the Arabian Sea. And in this Peruvian study, they drew their summary figure where they don't even list denitrification as a source of N2 at all. They show nitrate and nitrite being consumed, but they don't use the word denitrification, and they imply or maybe come right out and say that anomox is the dominant process forming N2 in the Peruvian oxygen minimum zone, and these 2 groups have had a pretty productive and healthy disagreement and about exactly what's going on here. And the reason why you get different answers is because denitrification and anomox are different, and they are going to behave differently in different conditions. And these 2 papers, for example, were studying different parts of the ocean. So let me explain that in a little more detail. Let me spell this out to you. I've said a couple of times now that denitrification is heterotrophic, and I can't overstate just how important that distinction is to anomox, which is autotrophic. That is that denitrification is dependent on the supply of organic matter from the surface. If you shut off that supply, if you no longer have a lot of organic matter being exported to the surface, to the deep from the surface, then you will not have denitrification anymore. Denitrification really needs that flux of organic matter. Anammox does not need that flux of organic matter. It will continue doing its anammox thing as long as there's nitrate and ammonia, and there's basically always nitrate and ammonia, right? You almost never run out of those two things, unless you're being outcompeted by another species of bacteria for those things, but you're not dependent on phytoplankton to produce nitrate and ammonia for the most part, right? That's a simplification, but compared to denitrification, that's a very important difference. And so they're still producing N2 even when the denitrifiers cannot. I see here that my slide is a bit cut off here, but you can see, I can explain it, and I will give you my real slides where it will not be cut off. I think you can mostly see here that, therefore, you will get different answers to the question of whether denitrification or anammox is more productive depending on when you take the sample that you're measuring. If you take a sample just after a whole bunch of fish pooped, then you will see a lot of denitrification. If you happen to take your sample when there haven't been a lot of fish swimming around, then you will not get much denitrification. So denitrification is very episodic. It goes in pulses depending on the pulses of the organic matter from the surface, which is inherently episodic, right? Organic matter is not constantly produced. You get big fluxes of it, big pulses of it, sporadically over time. And it also depends on whether when you take your water sample, are you filtering out large particles? So almost always when we take samples of water from the ocean, we filter that water through some series of filters to fractionate the sizes, because the ocean is dirty, right? And so if you're studying the microbes, then very often you want to filter out all of the large stuff. You want to get any small animals out of the way, you want to get fish poop out of the way, and focus on just the microbial fraction for the most part. But if you don't do that, if you keep the fish poop in your water sample, then you're going to get more denitrification. If you filter a lot of the large particles out, then you're going to get more anammox. So that's another reason why these groups have had different results from their studies, is because it really matters how you take the sample. Okay, so this figure I mentioned very briefly in the previous video. It's from this really good review paper that I highly recommend if you want to study this more. And so I just wanted to highlight where denitrification and anammox would be in this diagram. So denitrification is carried out by multiple kinds of bacteria that are doing lots of different things. In this diagram, you see that it is dependent on organic carbon, consuming nitrate or nitrite, and it's producing N2 gas. And in this diagram, N2 gas is leaving the system as a gas. Anammox is up here. It's only carried out by one particular group of bacteria, especially those in this genus Scalingua in the phylum Planktomycetes. And here it's shown ammonium nitrite being combined to form N2, which leaves the system. So this nicely shows how both anammox and denitrification result in N2. So I want to tell you one more story about the nitrogen cycle before we move on to other topics, and it involves places like this, like Starfleet Academy. So actually, Starfleet Academy was inspired or based on this actual building in California. You can see the similarities, which happens to be a wastewater treatment plant. So Starfleet Academy is a wastewater treatment plant. And so I want to tell you a little bit about how these bacteria we've been talking about in the nitrogen cycle are being used today by engineers to treat water. And the basic idea is that you can take advantage of the metabolic pathways of each of these kinds of bacteria to clean up our water. The basic idea here is that you take really, really dirty water. First, you use nitrifiers to convert ammonia from the dirty water into nitrate. This is an aerobic process, as we have talked about. The nitrate that you make from this process is consumed by denitrifiers in the second step to produce N2 gas, which can be effective in removing all of the nitrogen from the water as into gas, helping to clean up the water so it no longer is full of these different nitrogen species. And then you go back to aerobic conditions again for normal heterotrophic aerobic bacteria to consume any remaining organic matter in the system and release it as CO2. I honestly don't know why they're using NaOH here and HCl here. Something about the pH, I guess, and I'm not really sure what methanol is doing here. I know very little about wastewater treatment. Maybe some of you know more, and you can help us to understand this diagram better. But the basic idea here is by taking advantage of these metabolic pathways of nitrifiers and denitrifiers and heterotrophs, you can clean up our water using microbial processes. I also like talking about this because I can show these really cool images from this study taking microscope images of biofilm reactors in a wastewater treatment center that is doing the same process of nitrogen removal to clean up the water. And I like this because you can see in their microscope images the nitrifiers and denitrifiers and NMOX all in one biofilm together. So in these microscope images, they have used different probes to light up different kinds of bacteria with different colors. So this panel H is the best one, as well as panel G, where you can see the ammonia oxidizers. So that's the first step in nitrification. Out here in green, where they have access to oxygen. Here also you can see that it's on the outer edge of the biofilm. Below the nitrifying layer, you get these blue guys, which they've labeled, which is the color they labeled the nitrite oxidizers with. So that's the second step of nitrification. And then below that, you get NMOX, which is now in an anaerobic zone of the biofilm. So you get NMOX and the two steps of nitrification all living together in one biofilm, which is really, really pretty and showing how all those arrows that I was drawing in my PowerPoint slides, very often those arrows are going just a few microns between microbial cells because these bacteria can be very tightly associated with each other. For example, in particles in the ocean or as biofilms in wastewater treatment. Okay, that's all I have for you today. See you next time. Bye-bye.